Hey, it's Peter here with my FSHD. You know, we've been doing our research testing for FSHD since 2019. All right, so over 5 years. We've looked at more than 3,000 samples from all around the world. A lot has changed, but you know, if anything, we've gotten more accurate. Our testing is better than ever. And, but I still get a lot of questions. And I was looking at online on our site. I said, you know what? Some of our information is a little out of date. So you know what? Our reports have changed, our metrics have changed. We've really actually improved our understanding and the information we get out of all this wealth of data that we get out of everybody's sample. It's interesting for our research, but you know what? It might be interesting to you too. And I do want to always make sure that you understand exactly what's going on if you chose to get your results returned to you. So today I'm going to revisit a few things. First, I'm going to talk a little bit about FSHD genetics and epigenetics, just a refresher course, a little bit. And then I'm going to talk about the science behind the testing, so you understand what's going on if you're interested. I understand not everybody's interested. And then the third part, I'm going to go through the reports page by page so that everybody understands what's going on, what we're testing, what these results mean, that this is a research test, that sort of thing. But as always, you can contact me at peterjones@unr.edu, and I'd be happy to respond to your email and set up a time to have a phone call or a Zoom call and go over everything with you. But you know what? Sometimes you just need to see it multiple times, so here it is. It's going to be updated. Now, you know me. If you've talked to me on Zooms, I'll talk forever. So this does get a little long. It's kind of a long presentation because I want all the information given to you. But also I break it down into 3 parts. So if you want to, you can look through the whole thing. It's about an hour. Or just skip to the end, skip to part 3 to look at the how to understand your research report. Part 2, if you're interested in the science. And you know what? It's never a bad idea to get a brush up on the FSHD genetics and epigenetics. And that's in part 1. Okay, it's up to you though. We're just here to help you. We're trying to be responsive to the community and really appreciate everybody that has participated in our FSHD research testing program. It's just been amazing the way we've made it all around the world in over 70 countries. And you know what? We're going to make this the go-to test everywhere. All right, so thanks again for all of your help. And here we go. Okay, so let's start with some FSHD basics. All forms of FSHD are caused by genetic mutations that produce an epigenetic effect. And this is why I consider this ultimately an epigenetic disease. Okay. This epigenetic dysregulation leads to loss of repression and thus the gain of expression of the pathogenic DUX4 gene that's located on chromosome 4q35. So the key region of the genome for FSHD is the chromosome 4q35 D4Z4 repeat array. Okay, so you'll remember you have 23 pairs of chromosomes, so 46 total chromosomes, and you have one each from mom, one each from dad, so you have 2 chromosome 4s. And each chromosome 4, on the long end, the Q arm of the chromosome, at the very tip, there's what we call a D4Z4 repeat array. So a D4Z4 repeat unit, it's repeats of repeats. It's a highly repetitive sequence of DNA that is roughly 3,303 base pairs long. And this repeat is then repeated, head to tail fashion many times into what we call a repeat array. And this can be upwards of 120 or more repeat on chromosome 4. If you have less than 10, it's FSHD. Typically in the population, though, it's going to be 26 to 40 repeats or so. And since you have two different chromosome 4s, your 2 D4Z4 repeat arrays are most likely going to be different sizes. All right, so you have different alleles of your repeat array. All right. So FSHD is linked to this. It's very important, this repeat array. Now, there are other D4Z4s in the genome. And so here, these light blue, chromosome 10. Chromosome 10 is an almost near duplication of chromosome 4 repeat array. This is at 10q26. Now, this D4Z4 on chromosome 10 is 98% identical to chromosome 4. Okay, and then there's a distal region that we'll talk about in a second. But the key is that FSHD does not link to chromosome 10 most of the time. And there's a caveat to that I'll talk about. There's also other D4Z4s present in the genome at the acrocentric chromosomes, and they're just kind of scattered throughout the genome. And these are maybe 70 or 80% identical. Now the key is, if you're thinking about doing FSHD diagnostics or even just FSHD sequence-based research, you have to make sure that you're studying the right repeat, you're measuring the right repeat, and you're looking at the right repeat, because FSHD is linked to chromosome 4, not 10, and not these other ones. So it can get somewhat complicated. All right, so I mentioned there's a distal region. Okay, there is. So distal to the array is a subtelomeric sequence, and it comes in two main variations, and one's arbitrarily called A and the other is called B. Okay, and FSHD is exclusively linked to 4QA. Now we also have something we're going to talk about is 4AL, but oddly enough, the AL part, standing for long, is going to be here. It's not actually the subtelomeric part. Okay, so there's also a distal region on chromosome 10, 10QA, and 10 can also exist in an A or B. 10QA is not linked to FSHD. Okay, so now you have to make sure that you are now thinking diagnostically. Okay, what we want to know about is the chromosome 4q35 D4Z4 array in the context in cis, meaning on the same DNA sequence, linked to a 4A or 4AL subtelomere. All right, so why is this? Well, FSHD is caused by a pathogenic activation of expression of the DUX4 gene, and, well, the DUX4 gene is present within the 4q35 D4Z4 array. Okay, each repeat actually has a portion of the DUX4 gene. All right, and DUX4 encodes a cytotoxic protein. Not exactly sure how it ultimately leads to FSHD, but we know expression of DUX4 is bad in skeletal muscle. But the key is, at least part of the DUX4 gene is in each one of these arrays. The second part is going to be distal here, and we'll be talking about that in a second because that's really key. All right, so only contractions on 4A or 4AL and not 4B lead to FSHD. Okay, so I mentioned that these D4Z4 repeats can be present upwards of 120 repeats per chromosome 4, normally again in the mid-20s to 30s. A deletion in these number of repeats to between 1 and 10, okay, in the context of a 4A subtelomere, that is by definition FSHD1. Okay. Now the same deletion in the context of a 4QB, that is not FSHD, and on chromosome 10, again, not FSHD. We'll get to that because why that is. Actually, this has been a really key event. Now the other part about that is, you notice it says 1 to 10. If you have no D4Z4 repeats, you also can't get FSHD. And why is that? Well, the DUX4 gene, that is the pathogenic gene in FSHD, is located within the repeat. So, you need part 2, the subtelomere. You also need part 1. All right, so you need to have this combination in order to have FSHD. Okay, so now this is the key paper in 2010. It came out of Silver Vandermeer's lab. Key author was Richard Lemmers, who is the world's leading expert, in my opinion, on FSHD genetics. All right, this is great work. Stephen Tapscott, Rabbi Tawil, Runa France, the gang Sabrina Ciccone, George Padberg, Dan Miller. Anyway, you see the names. This is actually star-studded paper and really significant because this is what nailed down the answer to the controversy in FSHD as to what gene causes FSHD. People couldn't agree prior to this. Now, some will say that, we always knew. Well, maybe you did, maybe you didn't, but suspected. But this is key because what this showed is, it had been known for some years from work out of, again, from Richard Lemmers and Silver Vandermeer, that contractions on 4B never led to FSHD, but only contractions on a 4A. So why is this? Well, it turns out that there's the sequence that's key in 4A encodes a polyadenylation signal, all right, for the DUX4 gene, and this signal is not present on 4B. It's also not present on chromosome 10. And so what goes on is when you have the contraction and the epigenetic dysregulation, the DUX4 gene is able to be turned on. The RNA polymerase transcribes the DUX4 gene, makes the mRNA that is spliced and processed. And then you need this polyadenylation signal, this little sequence. It's a 5-base sequence that tells the transcription machinery to basically stop and add on this string of 15 to 20 A's, polyadenylation. And this is important because this stabilizes the mRNA and allows the mRNA then to be transported to the cytoplasm and bound up by the ribosomes and be translated into DUX4 protein, right? And then you end up getting FSHD pathology. In non-permissive subtelomeres, they don't have this sequence. So the DUX4 gene can be activated, transcribed, spliced, but you don't get this polyadenylation. So the mRNA is unstable, degraded, no DUX4 protein, no FSHD. All right, so this, and on chromosome 10, which also is not associated with FSHD, it's a single base, and that single base change on the 10A subtelomere is right in the polyadenylation signal. You don't get polyadenylation. So this was actually spectacular work. It not only explained why the contraction only on 4QA or AL, which are pathogenic and not on B, but it also really nailed down that it is the expression of the DUX4 full-length processed and polyadenylated mRNA that is what's absolutely key for FSHD. Okay, so I keep mentioning 4A and 4AL. Okay, you'll see it written like this: 4QA and 4AL. So the subtelomere is actually the same. It's this last little partial repeat unit that exists. Some people call this 4A short, and this is 4A long. But you see this little difference here. Right, we're just talking about this is the most common in the human population, and this is then next. It's going to be this little bit longer. Why does this matter? It adds a little bit more length for diagnostics, which is important. For sequence-based diagnostics, it's really important because you need to make sure that that you understand the sequence you're assessing. And it's also actually important for gene regulation studies, because if you're studying DUX4 mRNA, it does have a slightly different mRNA that it makes. And people have been misled by not understanding the sequence differences with 4AL versus 4A. But the key is they both are permissive. They both have the polyadenylation site. Okay, so that's actually what's key. Now this is what throws everybody off. Okay, you keep hearing me talk about permissive. All forms of FSHD require 4Q, D4, Z4 linked to permissive. What I mean by permissive is, 75% of the world population has a 4A or 4AL, at least one. 50% have two, right? And so why would that be? Because, 25% have two, 50% have one, and 25% have none. But you don't have FSHD. So it is not pathogenic. Having 4QA or AL, having the polyadenylation site, does not cause FSHD or lead to FSHD. You need other events to happen, but it's required for FSHD. So we consider this to be permissive for FSHD, where 4QB is non-permissive, because regardless of the other events that might happen in the region, if you're 4QB and you don't have the PAS, you're non-permissive, you can't develop FSHD. All right, so only contractions on chromosome 4 or 4AL or 4A 1 to 10. That's going to be FSHD1, and then also what's key is going to be that it really doesn't matter what your other allele is, and this is important because this shows you that FSHD is a dominant disease. One contraction on one chromosome 4 with a 4QA. That is actually causal for FSHD1, right? Doesn't matter whether your second chromosome's A or B, wouldn't matter. But then also FSHD2, which has other requirements, also requires 4QA or AL, at least one of the two chromosome 4s. It also has a specific requirement for a size of a repeat array. We're not going to get into that. But the other key in FSHD2 is you need a second mutation on chromosome 18 in the SMCHD1 gene, which is an epigenetic regulator of the region. All right? And that'll be important later. This also explains why you need DUX4, right? FSHD2, which has somewhat different genetic mechanism to get to the epigenetic dysregulation, still requires an FSHD permissive subtelomere. So again, it's key. Some of the permissive 4QA or 4QAL is key for FSHD. And it's going to be key for diagnostics. And I'm just sick of seeing these diagnostic reports that people send me that don't check to see whether you're a 4QA or 4QB, because there are people out there that have a contracted 4QB, and guess what? You don't have FSHD. All right, because if you have two 4QBs, you cannot be FSHD1 or FSHD2. All right, a contracted B does not lead to FSHD, and a contracted B in the context of an A on a healthy non-FSHD chromosome is also not FSHD, at least not FSHD1. All right, so in FSHD2, BB, you also cannot have FSHD2. However, if you do have an SMCHD1 mutation that could cause FSHD2, you can be a carrier of FSHD2. So while you can't pass on the chromosome 4 genetics to your offspring, your kids, you could pass on an FSHD2 mutation. So if your parents have FSHD2, one of your parents, you could be a carrier and pass FSHD on to your children, even if you're seeming otherwise healthy. So it's important to get this checked out as well. And actually the epigenetic testing we do checks this out. All right, and why is this again? Again, permissive. All right, DUX4 on both of your chromosome 4s in the context of a 4qb, they're transcribed, they can be spliced, cannot be polyadenylated, unstable, degraded, no DUX4 protein, no FSHD, both chromosome 4s. Doesn't matter if it's FSHD1 or FSHD2 that you're thinking about, you can't have either one, or even FSHD3 that's coming down the path. All right, so your FSHD chromosomes, FSHD1 is going to be linked. All right, chromosome 4. Now, I told you, I said it's exclusive to chromosome 4. I was very careful. Exclusive to chromosome 4qa or AL, D4Z4 repeats. Because there is actually a chromosome 10 FSHD1. But it's only if this is a hybrid chromosome that has a chromosome 4 D4Z4 distal unit and a chromosome 4 subtelomere. All right, so how does this happen? Well, it turns out, I mentioned that the D4Z4 and chromosome 4 and chromosome 10 are 98% identical. All right, well, sequences, especially repetitive sequences at certain points during development, very early in development, whether sometimes in spermatogenesis and oogenesis, and even at 1 and 2 cell stages, you can have a little bit of recombination, rare event. But what happens is you get a recombination between the very similar sequences. And this leads to a hybrid chromosome. If you had a contracted 10, which is not pathogenic, and a healthy 4, they recombine, you end up with a contracted 10 that has chromosome 4, D4Z4, at the distal tip, and the permissive 4qa or AL. It could happen with AL as well. And that actually is FSHD1. All right, and the key is here is this is often misdiagnosed by southern blotting and optical genome mapping. There's up to like 5% of FSHD1 cases are this hybrid chromosome. All right, now sometimes it does get picked up. It all depends on how many repeats. This could be 5 chromosome 4 repeats and 1 chromosome. It matters, but it can be missed. So if someone is clinically FSHD, and this does actually happen where really good FSHD clinicians, really fantastic neurologists that really understand FSHD will come to us and say, you know what, this person, I know they have FSHD, but the genetic test came back negative. Can you take a look? And we'll do our epigenetic test, and you'll see in the end that we're actually able to pick this up. And I can say, you know what, I bet they have a contraction on a chromosome 10. Yep, they got a contraction on chromosome 10. You know what, it's a chromosome 10-4 hybrid. The epigenetic diagnostics can actually pick that up. The key is we just want to help people get it right, but this is something that's got to be careful for. All right, so FSHD2, this is going to look otherwise like a not FSHD chromosome. Okay, 4qa, but you have between 8 and 20 repeat units. The key is you need this a pathogenic mutation in the SMCHD1 gene. Now, there's probably 20 different genes that can lead to FSHD2. More than 80% of the time, they're in SMCHD1. Very rarely, they're in other regulators, DNA methyltransferase 3b, LRIF1, and actually we haven't found the others. But most often it's SMCHD1. Regardless, the effect is going to be an epigenetic effect here that we're able to pick up with our epigenetic diagnostics. All right, so then there's the not FSHD chromosomes, greater than 10 repeats, doesn't matter whether it's a 4A or 4B, contracted 4B, contracted 10, and of course all the other D4Z4. Now, I throw these in here because, the sequences are similar enough that as people are doing sequence-based diagnostics, there's actually a proposed diagnostic out there that picks these up and actually gets it wrong, because the thing is, these D4Z4 repeats are not epigenetically dysregulated in FSHD. These actually are epigenetically dysregulated, all right? So if you're looking at epigenetics or any sequence-based diagnostics, you got to make sure that you're not studying just the garbage repeats that aren't associated with FSHD at all. Just got to be careful. That's actually a key with FSHD. The devil is in the details on everything FSHD is. A complicated disease. Gotta pay attention. All right, so what about diagnostics? Well, mention that the definition of FSHD1 is 1 to 10 repeat units. Well, the original diagnostics, and this was all figured out in the Netherlands, and again, the Netherlands is kind of the home of FSHD research and diagnostics and almost everything FSHD. Some people might disagree with me, but I think pretty safe to say that. What do is a physical measurement of the size of the D4Z4 repeat. That's traditional FSHD genetics. Just we know that this is 3,303 bases, and we know that 1 to 10 leads to FSHD. So let's just measure the DNA. All right. Now remember, you have the chromosome 10, D4Z4, and you're going to be now you have to distinguish. You're going to have chromosome 4 and chromosome 10. You have 2 of each, so you're going to have 4 sets of D4Z4 repeat arrays to study. You're going to be looking for a short one on 4, only with A, right? And FSHD2, well, measuring it's not going to help you a whole lot with FSHD2. All right. So the key is going to be that there are these things called restriction enzymes, and one of the important ones we're going to use is called EcoR1. These are isolated from bacteria. It's kind of a genome defense system in bacteria. So E. coli strain R, enzyme 1, will digest DNA of the sequence GAATTC. So remember, there's 4 bases of DNA, GATC. And so 4 to the 6th, it's a 6-base cutter. Every 4,096 bases on average in a genome, you should have an EcoR1 site. Now that's going to be skewed because this is a repeat. So you isolate this from the bacteria, and we use this. And so you're going to isolate blood from a patient, and then get the genomic DNA. You need very good high molecular weight DNA. DNA is actually pretty fragile and will break. You want to get this whole region intact. And here there's an EcoR1 site in the 4A subtelomere, and there's an EcoR1 site flanking this on the centromeric side. And we know that this is how much DNA there is here. It's about 5,000 bases and then a few thousand down here. And so we know that echo to echo is about the greater than, say, 40 kb is going to be more than 10 repeat units, and that's healthy. All right. Now the thing is, you have that exact same setup here on chromosome 10. All right. And then you're going to see it's going to be much smaller when FSHD1. And again, FSHD2 is going to be kind of hard. So you're going to cut with echo. I call it, some people will say ECOR1. I call it EcoR1. We're going to cut it with EcoR1. You're going to get this, and here you got, so all these types of repeats. So how do you figure that out? Well, it turns out there is another restriction site that's going to be in there that's going to be very useful, and that's going to be one called BLN1. You see this little B here. Okay, now the B, I told you that the chromosome 10 and chromosome 4 are 98% identical. Well, it turns out one of those spots where they're not identical actually is a different restriction site, and you can see that that's going to be cutting here, and we're going to be able to distinguish chromosome 10 from chromosome 4. All right, so we're going to cut with EcoR1. First, you just do EcoR1 by itself. That's going to give you all 4 arrays. Then you're going to cut with EcoR1 BLN1. All right, and so that's going to cut here at the B. It's going to cut here. It's going to cut here. So all 4 are going to get shortened a little bit. And then the chromosome 10 ones are going to get totally chewed up. They're going to get a cut every place. And that's going to not totally chewed up, but they're all going to be about 3,000 bases. And this is going to drop, be much smaller in size, and somewhat disappear, because your tag is actually, the tag that you're going to be probing for is just right here. So basically this is going to disappear. So what you'll be left with are 2 chromosome 4 arrays. All right. So what does this look like? Well, this is, we're going to use a technology called the Southern blot. All right. Now, you're a little confused. The Southern is was Dr. Southern at Edinburgh University. I think it's Edwin. I think his name was Dr. Edwin Southern. So that's why it's called the Southern blot. In 1973, if I'm correct, developed this technology, I believe, mostly for paternity testing. Which how do you track chromosomes? And so it's got nothing to do with FSHD, But the southern blot technique is, kind of depicted down here. What that is, the thing you have to remember is this cartoon looks really easy, right? You have to find your chromosome 4 and your chromosome 10s. Well, remember, there's 3 billion base pairs of DNA, on the haploid genome, so 6 billion bases of DNA, that are also getting cut by EchoR1 or EchoN-BLN1, right? And you have to somehow be able to find the specific sequence you want in the background of the rest of the human genome. All the other chromosomes all cut up, and they're all in the same tube together. So how are you going to do that? Well, the way you're going to do it is cut with your restriction enzymes, and then you're going to separate the DNA fragments by electrophoresis. So DNA is negatively charged, so it's going to run towards the positive, and you run through an agarose gel. Agarose is isolated from kelp, actually. I guess you can eat it even. It's kind of like gel. And based on the percent that you run, you're going to separate the DNA fragments based on size. And the smaller ones move through the gel much faster, and the big ones get held up. And so you end up, now you're separating all the DNA based on size. Okay, you're also going to run a marker that tells you exactly the size. So now you have a setup. You have all your DNAs that are cut with EchoR1 or cut with EchoR1 and BLN1, separated by size, but you still can't find them. Right? Well, then the next thing you're going to do is you're going to need to somehow have a probe. And figure out how to find it. So you're going to, the key to the southern blot is you're going to denature this DNA, okay, with sodium hydroxide. Double strand DNA becomes single strand. And high salt is going to be stable as single strand. You're then going to transfer this to a nylon or a nitrocellulose membrane, essentially like a piece of paper. All the DNA is going to come out of the gel onto a piece of paper and stick there and stay there. All right, but single-stranded, which means G can bind to C and T can bind to A. And so if you know the sequence you want to look for, you then take a radioactive probe and you're going to label it, make it single strand, and it will find the sequence that specifically you're looking for and make it radioactive, expose it to film, and you get an autoradiogram that it will show you the sizes of the DNAs that you're looking for. All right, and that's basically what southern blotting is going to be. All right, so let's go through it. All right, you're going to cut with EchoR1. You get your four things here. All right, now the key is we got a probe. Okay, this is P13E11. This was actually the key. You had to find something that was unique in the genome that's only going to pick up these 4 arrays, and that's what this probe does, P13E11. You radio label it, okay? And so you have your EchoR1 fragments radio labeled. Now when you do the southern blotting, you're going to get something here that's maybe 140 kb, kb being 1,000 bases, so 140,000. This one looks like it might be 110,000. Maybe this one's 60,000. And this one here looks like maybe it's 35,000. Okay, so you got a short one. Now you don't know if this is chromosome 4 or chromosome 10, but you got a short one, and that's interesting. So now there's the potential that you have an FSHD1 situation. All right, so now you're going to go with EchoR1, BLN1. You're going to cut with EchoR1, you're going to cut with BLN1, and you're going to now go through and you're going to look. And what's going to happen is you do your P13E11 probe. Now, the chromosome 10s are running off the gel. They're actually not even on here, right? And you're only left with the chromosome 4 EchoR1 BLN1 fragments, right? Chromosome 4, chromosome 4. So this short one, was actually chromosome 4. So now you know you have a short chromosome 4. Okay, so that's good news. Now, what you don't know at this point is if it's 4A or 4B, because guess what? B also has a little element, an echo site down here too. And so you have to do another southern blot. You should do another southern blot if you're a good lab. Now, sometimes they don't, but you come back and you do a second probing for 4A or 4B, and you can determine whether or not it's the short one's an A or B. Oftentimes that's skipped, unfortunately, and it can lead to some misdiagnosis. All right, so there you go. That's what's going on. So what about this situation where it's chromosome 10? Cut with echo. All right, the situation here, echo, there's your 4 sites. There's your short one. You don't know if this is 4 or 10. You cut with echo and BLN1. Only chromosome 4 is left. The 2 big ones are chromosome 4. This short one was a chromosome 10, and so that is not FSHD unless it has that distal element that maybe is a recombination that would have been missed on this test. Okay, because that recombination, if it's missed, think about it, the probe is here, right? The probe is going to be up here, and it only goes to the first cut. So unless the recombination was a complete recombination, that probe is not going to pick up, even if the fragments are not digested in that chromosome, in that hybrid. All right, so that's where the misdiagnosis comes in with the southern blotting. All right, now there's newer techniques that are out there. They have the same sort of needs, meaning you need high quality, high molecular weight genomic DNA, because these new techniques are going to do the same thing as a southern blot. It's kind of like automated, though. They're going to measure the size of the D4Z4, and they're going to detect whether you're a 4B or a 4A. They're going to use fluorescence. All right, so molecular combing, and I think the company may not be doing so hot. The only place I knew they were doing this was France, and I think they stopped doing it. But regardless, just so you're aware, if you've had this before, so green is the a probe that interacts with the D4Z4, hybridizes with D4Z4. And so you can measure how much green you have, and that tells you how long the D4Z4 is. And then you have specific colors that tell you whether you're 4A, 4B, 10A. And from what I understand, when this works, it works really well, but it didn't work very well very often. Anyways, my understanding, don't get mad at me if you're with the company, technology is kind of cool. But more recently, there's single molecule optical mapping, and BioNano has put this out, and this is seems to be what's kind of going more common in diagnostic labs now around the world and in the U.S. And so it's somewhat similar. You actually are going to nick the DNA and put fluorescent probes in, and you're still going to measure the 4QA, D4Z4, AL. You're going to measure chromosome 10, 4B, 410, 10A. So you can measure, and you get a lot of information here too. Find recombination of genome. Now, this can still, and does still, miss the 410 recombination, or the 10 chromosome 10-4 recombination that can be pathogenic, and I still get some of these. But in general, this is the same idea. You're measuring the size of the D4Z4. You need high quality, high molecular weight DNA from blood, fairly automated. The southern blotting takes a lot of skill. Leiden University Medical Center is probably the best in the world. The University of Iowa Diagnostic Lab is really fantastic as well. But I believe they're also switching to to BioNano. And then the last one that's coming along is this thing called long-range sequencing, put out by Oxford Nanopore Technologies, where you literally sequence through the entire region, and you start with a 4A, and you sequence through the whole D4Z4 and come out the other side, and you're able to tell how many repeats are there, any mutations. You also get some methylation, some epigenetic stats. Again, though, you need high molecular weight genomic DNA from blood. This is not as straightforward as it looks in this cartoon. There are some technical aspects to it that are a little tricky, but it does work. And it doesn't work in all sizes, but anything under 1 to 10 repeat units you'll be able to get. You get a little trouble maybe some of the larger FSHD 2-1. Although I've seen data up to 24 repeats, so it's possible. And now the technology for the sequencer is actually coming down in price. Now, the key is these top two, so the three of these technologies don't change the game from southern blotting in the sense that you still need high molecular weight blood DNA that's properly processed and very specialized equipment. All right, the price of these technologies has not come down, so you haven't increased accessibility, and you're not able to do it on saliva DNA. Now, the price of the nanopore sequencing may be coming down because they have different little portable sequencers and stuff. We'll see, but that's not quite being used as a diagnostic yet. All right, now what we do is something a bit different. So all forms of FSHD are caused by genetic mutations. We focus on the epigenetic effect, right? All of those are looking at the genetic effect. We're looking at the epigenetic effect, all right? And so epigenetically, a genetically not FSHD chromosome 4 has a epigenetic status that is epigenetically repressed, transcriptionally silent, for the most part, and highly methylated on the DNA. And it doesn't matter whether it's a 4A or 4B, right? FSHD1, you have one chromosome 4 that is epigenetically on, unmethylated or low levels of methylation, and the other one is healthy levels, again, methylated and off. And in FSHD2, both chromosome 4 D4Z4 arrays are epigenetically dysregulated. The yellow means there's no methylation or very low methylation. The black means high methylation. So if you just think epigenetically, and you go through and you look, you have methylated, methylated, unmethylated, methylated, unmethylated, unmethylated. All right, and that's the basis of epigenetic diagnostics, is this distinct FSHD epigenetic profiles. All right, so, right, I guess saying it again, epigenetic diagnostics is based on these epigenetic profiles. All right, so what are we going to do? Oddly enough, the first thing you ought to do, the first thing we do, is something that's a genetic analysis. First thing we want to know is, do you have an FSHD permissive 4A or 4AL? We don't care whether it's on or off, methylated or not. We want to know, do you have one? Because if you're BB, we immediately know it's not FSHD. And the second basis of that then is that our epigenetic assay is based on a permissive chromosome 4. We don't care about the epigenetics of a 4qB distal repeat unit. We look at the epigenetic status of the last D4Z4 repeat unit in the context of a 4A or a 4AL, because this is actually the repeat unit that leads to DUX4 expression and is the pathogenic repeat unit. All the other ones, who cares? This is the one that matters. All right, so we're going to say, what is the epigenetic status of 4A or 4AL, right? And then the next thing we're going to look at is, okay, if you're FSHD, is it FSHD 1 or 2? And we're going to look at the epigenetic status now of all the D4Z4 repeats in chromosome 4s, and you can see how very quickly you'd be able to pick it all up on whether it's FSHD, then it's 1, or FSHD 2 by how many methylated repeats there are present. In FSHD 1, most of the repeats are still methylated, because the small, think about this. If this is 50 repeats long, and this is 5 repeats long, less than 10% of the repeats are epigenetically dysregulated, right? So you're just going to be mostly methylated, where in FSHD2, they're all dysregulated. Okay, so that's the basis of epigenetic diagnostics, right? And the key is it can be performed on low-quality saliva DNA, because these are all small. We're not measuring the size of the array. All we need is little 500 base pairs pieces of DNA, low-quality saliva DNA that can be collected through the mail anywhere in the world, stable for years. You can actually pull DNA out of the freezer. We've assayed DNAs from the 80s that have been in the freezer for 30 years. Is that right? 80s, how far? Yeah, I guess like 30 years. Before there was a diagnostic for FSHD, and we can actually test those for their methylation state. You can't do it with any of the other technologies because they're fractured. Defreezing DNA is really bad for genomic DNA in respect to keeping it the size and the integrity. All right. Now, one thing I want to bring up is we focus on epigenetic in the FSHD. There are genetic definitions of FSHD, right? So by definition, FSHD1 is 1 to 10 repeat units. 9 repeat units with a 4A, 10 repeat units with a 4A, 11 repeat units, and you're not FSHD. Now, even if you're clinically FSHD, you won't be considered genetically. You come back as a negative, all right? Now, we look at it slightly differently. I would look at it and say, if you're epigenetic, if you're clinically FSHD, I don't care whether you have 9 repeats, 10 repeats, or 11 repeats. These situations would come up positive by epigenetic testing. These would be negative by epigenetic testing. I actually believe that these are and should be considered positive. And so sometimes people get a result through the MyFSHD testing or through my lab's epigenetic research testing where we give them you're positive for FSHD, and they get a negative result. But then I find out that they have 11 repeat units or 12 repeat units. If you're clinically FSHD and you're epigenetically FSHD, I don't care what your genetics are. You should be FSHD. All right? And that's kind of what's going on here. We get this conflicting FSHD epigenetic and genetic results, and this causes some stress with clinicians and patients. Now, I don't have any patients because we're researchers and we just have subjects, and we only do a research test. But if someone is clinically FSHD, but they're not genetic FSHD1 because they have 11 to 14 repeat units, they're not genetic FSHD2 because they don't have an SMCSG1 mutation, they may have FSHD2 levels of methylation for various reasons, but no mutation, but they are positive for epigenetic FSHD1, situation like this, where one chromosome 4 is 11 repeats, 12 repeats, but epigenetically on. The other chromosome 4 is pretty close. I'm kind of epigenetically on, kind of off, maybe don't really know. If you're clinically FSHD, and this is the situation, I think it should still be considered FSHD. And the problem is these individuals, they're going to be searching for what is their disease. They're going to be doing other diagnostic testing. They're not going to be able to get into clinical trials. They're not going to be able to get therapies. And we got to fix this, because I believe epigenetic FSHD and the combined with the 4A, 4AL is actually combined with clinical FSHD should be considered FSHD. All right, so now we all understand FSHD genetics and epigenetics, right? And the differences between FSHD1 and FSHD2, and non-FSHD, right? The FSHD region is epigenetically repressed in most individuals, or at least individuals without FSHD. In FSHD1, the signal that tells the cell to shut off this region, the number of repeats, which are recognized as a region of the genome to be silenced, that's actually lost. The signal is broken due to the contraction, and the cell does not efficiently recognize the region to be epigenetically silenced, and so this is FSHD1. You get aberrant expression of the pathogenic DUX4 gene. Right? In FSHD2, the cell still recognizes the region and says, yes, this is a region we should shut down, but the mechanism or the machinery to shut off the off switch is actually broken, right, due to mutations, most often in an SMCHD1. Right? This is a component that's required for epigenetic silencing of these types of regions of the genome. Right? The end result is the same. You have aberrant expression of the DUX4 gene in your skeletal muscles, so it doesn't matter whether it's FSHD1 and the signal is broken, or an FSHD2 where the switch is broken. You still end up with FSHD, right, due to DUX4 expression. So how do we translate this into an epigenetic a diagnostic? And of course, right now it's a research test. We'll show you hypothetically how it could be used as a diagnostic. This was invented by Takako Jones, my partner in the lab, and we're going to take you through in this section exactly how we do that. It involves epigenetic sequencing, right? Actually, on a base-by-base DNA sequencing mechanism to determine the methylation status of the cytosines in the region. Okay, it's called bisulfite genomic sequencing. We do a bisulfite genomic PCR to amplify up these specific regions, and then we're going to sequence them. And there's two different ways we do the sequencing. So I'm going to go through all of that. Every way that we do it is incredibly accurate, over 99% accurate. Some are faster than others. Some different techniques are cheaper than others, which is why we do them. But the important thing is we're able to do it on saliva DNA, or actually any DNA really that we can isolate from you, and low cost. We can do it pretty fast, and this is how we're going to get an extremely accurate result to you as a research test. And then we'll cover that though in the third section. So right now, here's how we do the testing. Okay, so first thing we want to remind everybody, this is a research grade test that we do through my FSHD and the Jones Lab at UNR. Okay, that means the result is not considered clinically relevant diagnosis, or it's not actually even considered a genetic test in the USA. All right, and also this result may not meet clinical trial inclusion criteria, although this is a little sketchy. It may. It depends on the clinical trial. But if the goal is to get into a clinical trial, if our result comes back positive, it's quite possible you're going to have to go and take a CLIA-approved test to confirm the results that we have. And you know what? You should do that anyway. All right, so distinct epigenetics in FSHD. If in non-FSHD cases, you have these expanded D4Z4 arrays. They are epigenetically silenced. It means they're heavily methylated. In FSHD, due to the contraction or the deletion in the repeats, which are recognized by the cell as being something they want to silence. I mean, that's basically what's going on. The repeats are recognized, repetitive DNA, as this is kind of bad DNA. Let's silence it in somatic cells, and it's going to be epigenetically silenced. The deletion of the repeats, now the cell doesn't actually recognize this as necessarily a repeat, and it's going to be on a cell-by-cell basis. But is this something that should be silenced or not? And, enough of the alleles are unsilenced and epigenetically on, so they're not methylated, And so that's FSHD1 when you have between 1 and 10 repeat units. Now in FSHD2, you still have a healthy or non-FSHD size repeat array on both of your chromosome 4s. So the signal that says turn me off is still there, but the off switch is broken, and that's due to mutations in other genes that regulate the epigenetic status of the region. And because it's the off switch that's broken, both alleles of chromosome 4 are actually epigenetically dysregulated. So you have methylated is not FSHD, unmethylated is FSHD1, and unmethylated is FSHD2. Okay, so how are we going to do the test? So the first thing, oddly enough, we're going to do for an epigenetic analysis is a genetic test. So we're going to try to figure out if you are FSHD permissive, meaning do you have an FSHD permissive chromosome 4A or 4AL subtelomere? All right, and if you don't know what I'm talking about, you need to see the earlier part, part 1 of the presentation. Okay, because FSHD is only associated with 4A and 4AL subtelomeres. Okay, so we're going to check you out, and 25% of the world population is going to be 4B, meaning not permissive for FSHD, so you can't have FSHD if you're 4B. 75% of the world population, roughly, has at least one 4A or one 4AL, so is permissive for FSHD. So they have the potential to be FSHD. It doesn't mean you are FSHD. These are not pathogenic. Permissive, okay? So 25% of the world is non-permissive. 75% is permissive and potentially could be FSHD. All right, so what are we talking about? So we're going to go through, and this analysis is going to be called haplotyping. Okay, now what is a haplotype? Haplotype is a set of genetic elements, or genes, if you were to say, that tend to be inherited together. So the FSHD region has your D4Z4 array, your distal subtelomere that determines whether you're permissive or not permissive, and the upstream on the centromeric sides are these simple sequence length polymorphisms that we're going to use to track chromosomes through your family, and also to help us understand whether you're permissive or not. So the first thing we're going to do is a PCR, polymerase chain reaction, a genetic test that is specific for 4A, 4AL, and for B, and find out what are you. Do you have an A, an AL, a B, two A's, two ALs, AB, whatever. We'll figure out what your subtelomere is. We're also going to figure out what is your SSLP, because your SSLP is often linked to your subtelomere. It's a haplotype. They're inherited together. What these are really short repetitive sequences that are highly variable in the human population, but there are specific ones in this region that are associated with certain FSHD haplotypes. For example, 161 often goes with 4A or 4AL. So you'll often see people are 4A 161. And when we see that, we know that they're likely FSHD permissive. All right. Chromosome 4B often is 163 or 168. If we see someone who's 4B and two 163s, 168s, we know that they're non-permissive. Okay, so this is all just putting it together to characterize your FSHD region. All right. So then the next test we're going to do is an epigenetic test, and the whole basis of this is epigenetic DNA sequencing. So there's 4 DNA bases, C, T, A, G. However, C can exist in a methylated form in humans. Okay, and so methyl C is, sometimes they consider it like a 5th base. Methyl C in the context only of a CG dinucleotide. So here's actually the one to focus because this cartoon was not made correctly. CG in humans at least, and in other organisms you can have non-CpG, but in humans you only have CG dinucleotides that can be methylated or unmethylated. And the reason you do that is CG on one strand is CG. Remember, DNA is directional. 5' to 3' here goes 5' to 3' here. G binds to C, C binds to G. So you have a symmetrically methylated region. Okay, and this is what we're assaying. So there's a chemical called sodium bisulfite. And what sodium bisulfite does is it converts C's that are not methylated, like this C right, C right here, is not methylated. That becomes a T, which is actually read as a U, like right here. All right. C's that are methylated remain a C. All right, so it's a chemical modification of the cytosine to make it into a uracil. And so when you do DNA sequencing, anytime you see a T, okay, because where there should have been a C, you know that that C was unmethylated. Anytime you see a C where you should have seen a C, you know that C is methylated. So sodium bisulfite converts C's to T's, while methyl C's remain C's. And by doing this bisulfide sequencing, you're able to get the epigenetic state of the DNA. All right. So again, you see here you have CC, right? The C is unmethylated. That's a U. That is a T when you finally do the sequencing. This C was methylated. That stays a C after sodium bisulfite conversion and is a C in the sequencing when you do the methylation, when you do the sequencing analysis. And so again, you're getting the base by base methylation sequence. Okay, and this is what we're doing with our epigenetic sequencing. All right, so where are the regions we're going to look at? Well, it seems very complicated, but remember you have chromosome 10 D4Z4s, chromosome 4 D4Z4s. All right, you can have a subtelomere that's a chromosome 10A, 10B, 4B, 4A, 4AL. There's also this funky one called 4A166, which we'll talk about in a second. All right, and FSHD 1 and 2 are only associated with 4A and 4AL. So what are we going to do? Our first epigenetic assay is going to look at this region right here and see what is the epigenetic status for 4A and 4AL. All right, if you have them. If you don't have them, then it comes up blank. All right, then the next thing we're going to do, because this will tell us FSHD or not FSHD, right? Unmethylated, unmethylated, methylated, methylated. And then we're going to look at all the D4Z4 repeats. That's going to pick up chromosome 10, chromosome 4, even if it's a B, healthy chromosome 4s. We're going to pick everything up, and this will tell us FSHD1, FSHD2, or carrier of FSHD2. So this is the basis of the assay. This is kind of the flow chart. We're going to prepare your genomic DNA from saliva, from blood, from cells, pretty much from anything. Okay, bisulfite convert, do the 4A BSSA, bisulfite sequencing for A, BSSL, bisulfite sequencing for L, and this is our first assay, and we're going to do this on everybody. Actually, even if you're BB, we're going to do these assays just to make sure that there's not something made a mistake, just to make sure there wasn't a mix-up, and, we like to, they should come up negative, no product, and, that's what happens. We always like to double-check ourselves. It's a research test, but you know what? It's still somebody's results. Okay, so epigenetic assay 1, if you have high levels of methylation, we know that's not FSHD. Low levels of methylation, different metrics, it is FSHD, or it's associated with FSHD, okay? Now, 4A and 4AL have different metrics, okay? 4AL is heavily methylated even in FSHD, so we actually have different metrics to look at. And this is important, and this is one of the several places where other methylation assays in the FSHD that are being attempted to be used for diagnostics get it wrong. Okay, you need to distinguish 4A from 4AL. All right, so then we're going to go through and do our next epigenetic assay. We call it BSSX, and we're actually going to do this even if you're not FSHD. Again, we do every assay, but this flow would be if you're FSHD, are you FSHD1 or likely FSHD2? High levels of methylation across all the repeats is FSHD1. Low levels across the non-contracted array are FSHD2. All right, so that's kind of the flow. All right, so how accurate are these results? Well, we have two different ways that we do the DNA sequencing, and both of them have shown to be greater than 99% accurate to identify FSHD versus not FSHD on genetically defined samples that we have in the lab. And we've actually together done more than 700 samples, and we'll show you the stats in a second. But the two methods are the old school Sanger sequencing method, and this was invented by Sanger, and we've been using this since 2014 when we started doing this technology on a research basis. We still use this today for individual samples. It's very quick. We can do this in 2 to 3 days. It is more expensive for us, so we tend not to do this, but occasionally if people have a doctor's appointment they want to bring their results to, if they're just really freaking out, or if we need to get some quick confirmation, we still do this. It doesn't require any special equipment, just a PCR machine and very standard stuff in any lab. More recently, though, we've moved in 2021 to what's called next-generation sequencing. Now, others have published on this that are copying our technology, but we've been doing the next-gen sequencing since 2021. In this, you have a microchip where you can run batches of 70 to 96 or even more samples, depending on how many barcodes you have. This takes a little bit longer, but the reason it takes longer really is because we need to get 70 to 96 samples sent to the lab from all around the world. And, we can get anywhere between 5 samples to 20 samples in a week. You just never know. In the end, it costs the same amount to run one sample as 96 samples. We found the good cost break for us is around 70 samples because it's $50 to $60 per subject. And so this is cost effective, so it's worth waiting to get this done. It also gets a lot more data than you get in this. You'll see this in a second. You do, however, need a next-gen sequencing machine. Now, we use a Thermo Fisher Gene Studio S5 with an ion chef to make the chip. We use this also because we do our own custom neuromuscular disease array, and we have other reasons we use it. But you could use a different, cheaper NGS machine if you wanted to. But we use the Thermo Fisher machine. All right, so how does this method work? I'm just going to take you through it. A little bit of nerdy, little kind of standard molecular biology. Sanger sequencing, we call this TA cloning. You do your bisulfide BSSA PCR or BSSL or BSSX, and you get a PCR fragment that you are then going to clone into a plasmid. This is a little circular piece of DNA that can be propagated in E. coli. And you transform E. coli, and each one of these white colonies here contains a PCR fragment that originated from one chromosome from one cell in your cheek. Okay, so each one of these came from a different cell that we isolated from your cheek. We got thousands and thousands of cells in your saliva sample, and each one has the same DNA sequence at chromosome 4. 2 chromosome 4s, right? The very distal end is going to be the same sequence in both chromosome 4s, but epigenetically they're going to be different. So we pick a series of these white colonies, 15 to 30 of them, and we have them sequenced. Again, each clone represents one chromosome 4 from one cell. They're all different, and we get the sequencing. And this just shows you the different color-based capillary sequencing. All right, so what does it look like? Well, we're going to look at 56 CPGs. Remember I told you just a second ago that in humans, only Cs that are in a CG dinucleotide can be methylated. They can be methylated or unmethylated. Red represents the methylated state, right? That's going to be a C that remained a C because it was methylated. The blue is going to be an unmethylated C that was converted to a uracil and then read as a T. And so we're now going to be able to go through and see methylated, unmethylated, 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 methylated, and then the whole thing. And so we get an epigenetic sequence here that shows 51.8% methylation. So in those cartoons, I show it as all methylated or all unmethylated. This actually is what you get. You get 51.8. Well, let's look at another one. Another chromosome also is 51.8. Okay, so that's the same. Oh, but look, the methylation pattern is different. Even though it's the same overall level, it's different. What about the next one? Well, now we're all down to 50%. Down to 44.6%. 41%. 33%. We're getting some different ones, right? Now, if we actually look at 25 different chromosomes from 25 different cells, all chromosome 4s in this individual, right? Each left to right represents one unique chromosome 4. Some are 3.6%, some are 51.8, and you have everything in between. So even though genetic DNA sequence is the same, the epigenetic sequence or the epigenetic state is different on a cell-by-cell basis. And these are cells from your cheek, okay? So now we're going to go through, we had these stacked purposefully. We didn't randomly get this order. We had them stacked, highest to lowest, or lowest to highest, however you like to think about it. And now we're going to look at quartiles. So we're going to separate it into the first quartile, and you're going to see this on your report, Q1. It's going to tell us that's 12.5%. Q2 is 24%. That's this right here, 50 halfway through. Q3 is 33.9%. That's right here. Now this individual has one 4A and one 4B. So we only assay 4A. We don't care about 4B. So our assay is specific for 4A. And so that means that the single chromosome that they originally inherited from mom or dad, the 4A, okay, is between 3.6 and 51.8 methylated. Okay, so that's a huge difference in epigenetic state from the same chromosome, okay, just on different cells. And since it's AB, we're going to look at Q2 halfway through, 24%. That is FSHD, probably a mild FSHD, quite honestly. Okay. Then we're going to go through and we're going to do, we know that BSSA is FSHD. We're going to do BSSX and look at all the D4s, E4s. Now we're going to look at 59 different CPGs, and they're 42%. That's greater than 30%, not FSHD2. This individual is FSHD1. Now let's go look at another individual, same sort of thing, right? 1.8% to 69.6%, almost 70%. But actually looking at this, it looks like there's two kind of populations. You kind of have a low population, and you kind of have a high population. This person might be AA. So we're going to look at our quartiles again. First quartile, second quartile, right here. Third quartile, right here. And this person is 4A, 4A. So now we're actually assaying both their short chromosome and their long chromosome. Now everybody's got a short and everyone's got a long. Just that both of your short and your long might both be healthy size, right? You could have a 20 repeat unit and a 50 repeat unit, okay? And the shorter one is going to be less than the, but you also could have a 3 repeat unit and a 50. We don't really know. We don't care actually how long your repeat units are. All we care is epigenetic state. Since they're 4A, 4A, we're going to use first quartile, 17.9. That is definitely FSHD. So this individual, this is 4A, 4A. They have an FSHD chromosome and a non-FSHD chromosome. Okay, a short and a long. That are both A. We're then going to do BSSX again. We got 53.4%. That is higher than 30%. Not FSHD. Again, another case of FSHD1. Okay. So how accurate is this? We've done 318 genetically confirmed samples. 315 we got correct. 3 we got incorrect. When we look at FSHD1 for FSHD2, we start to miss a couple of them. FSHD versus not FSHD is greater than 99%. FSHD1 versus FSHD2, they're still FSHD, but every once in a while, due to some unusual circumstances, an FSHD1 case can look like FSHD2, and we get it wrong calling FSHD2. On rare occasions, we call somebody FSHD2 when they're actually FSHD1. We can explain this, kind of later in the talk. So what about the next-gen sequencing method? The same idea. We do BSSA, BSSL, and BSSX, but this time we're going to add little special codes to each PCR product that are specific to the individual. All right, there's no cloning involved, there's no bacteria involved. We just get the PCR products that are labeled specifically. So if you are 4A, 4AL, we'll have 3 products for you. If you're BB, we only have 1 product for you. If you're AA, we'll only have one. We have 2 products, BSSA and X, okay. We then have these all in a tube, all right, and then Takako makes a microchip on the Ion Chef, and they all get arrayed on a microchip. Again, we have the barcodes to do 96, but once we hit 70, people are waiting, and we want to get the research results back to people. So we typically at least 70 at a time. All right. Then we put them onto the chip onto our genome sequencer, our GeneStudio S5, and you end up with all these barcoded BSSA, BSSL, and BSSX products. The computer sorts them all out, and then it's very important because we find all these interesting other sequences that other people are going to miss, right? So even though it's BSSA, you'll notice, well, there's a couple of other sequences that get read that are in there that are just kind of some garbage sequences. The computer sorts them out and gets rid of them. PCR is a little bit dirty. Now, instead of 25 reads, 2,800 reads, 2,800 different chromosomes. And what we do, the computer actually sorts out based on little sequence variations to know that all of these are unique. All right, so all of you are assaying 2,800 different cells from your cheek. All right, the low level is 12% methylation. The high level is 100% methylation. This person's AB. So even with a single chromosome A, you can be between 12 and 100. Okay, that's what the kind of data that this kind of depth gets you. Now, because of that, they're actually, though, we know we take Q2, 56.52. That is not FSHD, right? And then if we're going to look at BSSX, it confirms that it's still not FSHD, and it's definitely not FSHD2. But think about this range that people have. You think, okay, this is a healthy individual range. Everybody's a little bit FSHD. All right, now let's look at this case down here. We're going to look at, this is also 4A, 4B. So what's the range here? Well, now you see a lot of zeros, right? A lot of zeros coming in. And the maximum range, though, still is almost 80%, okay? 95%, 90%. You actually, 25% or more are healthy epigenetic states, kind of the chromosome. But on average, right, here at Q2, it's not really an average, but at the second quartile, it's 24%. That is FSHD. And we look at the BSSX, that's FSHD1, all right? And this time we draw 4,317 different chromosomes to get this level of depth. Okay, here 6,405 different D4Z4 repeats, not chromosomes, because now this is only specific to each repeat, right? So that's fewer chromosomes. All right, what does it look like? Here's kind of our heat maps. Remember, blue is unmethylated, red is methylated, and both individuals here are A, B, and these are the ones that we just showed. So the not FSHD, you see a lot more red, but you still see some blue. And in the FSHD, you see a lot more blue, but there's still some red. And this is actually what you are. This is the epigenetic variability that makes you different from your sibling or from your parent or from your child that has FSHD. Right? You have a unique epigenetic signature. This is only 100 cells that were randomly chosen by the computer. But think about this. You are the trillions of cells that make you you. They have the same DNA sequence of the region, but each one has their different. You have some that are 100% unmethylated and very on, but you have some that are methylated and very off. And in this mixture, and what muscles are these in, and how do these get individually activated? Everybody's epigenetically different. Even if you have the same numbers, the patterns are going to be different. The percentages are going to be different on an individual basis. Identical twins are different. All right, and this is the kind of data you get from the next-gen sequencing data when you assay it correctly. All right, this is that 50% mark, and just see you're 50% FSHD. 50% healthy, right here. It's not really, it's really a second quartile mark. Very clear that we're just assaying at this point. You get the non-FSHD versus FSHD. So how many have we done? We've done 463 genetically confirmed samples. We've been correct 459 times. And honestly, these 4 incorrect ones are pretty dang close to being correct. We'll get to that in a second. So again, FSHD1 versus FSHD2 is a little bit more complicated because it's unusual, but it's still FSHD, right? We're getting it right that it's FSHD. Maybe it's 1, maybe it's 2. Most of the time we still get that right, still greater than 95% of the time. But it is a little bit of a hole in the system. All right, so what happens when you get a conflicting result, right? If you're clinically FSHD and came to us for your research test result and came back and we said, yes, you are epigenetically FSHD, go ahead and get your clinically approved test. And then you go get your test, and it comes back negative for FSHD1 and negative for FSHD2. What's going on? Well, again, it turns out that, and this has happened several times, there are 2 possibilities. Well, there's 3 possibilities. One, we're wrong and we don't understand why. That is actually the rarest of all possibility. It could be a chromosome 10 rearrangement, right? That could be one, and we're picking it up, and we're able to figure that out because on your genetic test, you'll have a short chromosome 10. But what else could it be? Well, it turns out that if you have between 1 and 14 repeat units, by definition, the testing agency will come back and tell you you're negative for FSHD1. You have 11 repeat units. That's not FSHD, right? Your FSHD2 methylation levels might not be low enough to be FSHD2. You don't have an SMCHD1 mutation. So you don't fit any of the accepted criteria for FSHD, not 1 or 2, but you're epigenetically FSHD in this situation. I would make the case that actually should be considered FSHD, right? Because by definition, these situations are FSHD1, 9 repeat units, 10 repeat units with a 4A, but as soon as you're 11 repeat units, you're not considered FSHD, even if you're clinically FSHD and even if you're epigenetically FSHD, right? I believe it should be this situation where epigenetic FSHD and clinical FSHD go hand in hand. That should be considered FSHD. These situations here, for maybe genetic counseling, they can be considered FSHD. it gets complicated when you're thinking about inheritance and passing it on, but at least for therapy, for determining what the individual has as a defined, confirmed disease, and being able to get into a clinical trial, and being able to get therapies when they're available, I believe all of these situations should be included as FSHD.
All right, so now we're going to go through the report, okay? I get a lot of comments from people. The first comment I get is, well, so what is this telling me, okay? So I want to make sure that everyone has an opportunity to understand online. But of course, don't forget, you're always able to send me an email at peterjones@med.unr.edu or peterjones@unr.edu. It doesn't really matter. Not sure why we have the med in there. Either way, I'm going to go through it page by page, and hopefully, at least you'll have a resource to go back to understand it, okay? And one more thing. If you haven't received your report, I send them out. I don't know that you got it, actually, unless you contact me back and say, well, either thanks or I don't understand it or can I set up a call or whatever. So sometimes, months will go by and then suddenly I get someone that says, I never received my report. Well, check your spam. Check your junk file or whatever. But always feel free to contact me and I'll get it sent out to you. But also make sure that you had to have filled out our consent form. If you didn't fill out our consent form, we can't actually perform the analysis for you or return you the results. All right, so it does take about, 2 to 6 weeks lately, even sometimes as late as 8 weeks because we're getting overwhelmed with the number of samples. So it does take a while, but you're always free to check with me. But once you get it, log in here and let's go over the report. All right, let's get going.
Okay, so grab your report and we'll go over it. Now, if you have an older report from a couple of years ago, it may look a little different, but this is the current report that we're putting out. And if you'd like an updated report, I'd be happy to, update one for you because this has a lot more information in response to, a lot of suggestions from y'all and the common questions we get. So to start out, it's going to have your name and then the overall result of the analysis of your sample, okay? And so this is actually all you really need. What this says is Mr. Vandelay's sample, the total analysis came back consistent with what we find when we do the same type of analysis on a genetically confirmed case of FSHD1. So the analysis, we can compare against FSHD1, FSHD2, or not FSHD. This says it would fit into the FSHD1 category. We are not, however, telling them that he has FSHD1 or FSHD. And that's always a little bit confusing. It's a research test. All right, so section 1, then it'll be an overall narrative explaining your results. And I'll do my best in kind of lay terms. And it's complicated. FSHD is complicated. Science is complicated. I do my best to explain what's going on, though. Then the section 2 will be the experimental details of your analysis. And this is where it starts. And this will be the genotyping. Do you have an FSHD permissive chromosome 4A or 4AL? You get haplotyping results here. Section 3 will be the first epigenetic test, FSHD or not FSHD. And then section 4, everybody should have a result in section 4. Now in section 3, if you're BB, if you do not have a permissive result, we already know you're not FSHD in the test, and so this is not applicable. In section 4, everyone should have a result because we're going to find out, is it FSHD1 or FSHD2? And even if you're not FSHD, because you're BB, we can find out if you're a carrier of FSHD2. And it's also another confirmation of a not FSHD determination in section 3. And then section 5 will be a more detailed. I'll go over the numbers, where they fit in our ranking. Basically, how did we come to the conclusion based on the numbers? And when I keep saying I write personally. I do all the analyses. I write each and every report, and I send out the reports. Okay, so it comes through me. No one else in the lab knows your name. No one else knows anything about you. It's confidential, and I actually do this on my own, which is also why it takes a little while, because each report takes about 15 minutes. And if you think about it, I'm going to do 70 at a time when I get them, so it takes quite 12 to 15 hours to get them all done. But I do my best, and we try to get them out to you. All right, so how accurate is the research testing we do? Well, if you saw the previous section, we have 2 little variations on it, but overall we have done 781 genetically confirmed samples. We have been correct 774. We have an overall success rate of 99.1% at correctly characterizing the samples as either FSHD or not FSHD. And then when it comes down to FSHD1 versus FSHD2, we're still at 96%, but occasionally there's an FSHD1 case that looks like FSHD2 and an FSHD2 case that looks like FSHD1. It's complicated, it's epigenetics. The key is, though, regardless, they're still FSHD, and you'll be, and that's really the most important thing. All right, so what does a result of FSHD1 mean? Okay, so you'll remember that genetic FSHD1 is defined as between 1 and 10 D4Z4 repeat units on a 4A or 4AL chromosome. Okay, so this situation here, it does not matter what the second chromosome 4 is. It can be A, it can be B, it can be L, whatever. 4A or 4AL, and that is genetic FSHD1. All right, no, that's not what we look at. We're looking at epigenetics, right? So this is epigenetic FSHD1. One chromosome 4A or AL is epigenetically dysregulated and lack of methylation, unmethylated, and the other chromosome 4, regardless of whether it's A or B, is methylated and epigenetically off. All right, so we look at epigenetic FSHD. All right, so go to the case of Mr. Vandelay and FSHD1. What that means is this is the situation, right? One epigenetically dysregulated, unmethylated chromosome 4A, and one that is epigenetically off and methylated. All right, that's what it means. Epigenetic FSHD. We do not measure the size of the array. We don't know how many repeats there are. Honestly, don't care how many repeats there are. We care about the epigenetic status. Okay, so what does a result of FSHD2 mean? Again, genetically, FSHD2 is defined either through a neuromuscular disease panel for mutations in the FSHD2 gene, the most common FSHD2 gene, SMCHD1, or direct sequencing of SMCHD1, and actually D4Z4 DNA methylation analysis. Slightly different methylation analysis than we do, but the same general idea, looking at overall D4Z4 methylation. Really, they should do both, because some mutations that are found in these sort of surveys are actually called variants of unknown significance, and they are ambiguous. And so they don't actually know if they're pathogenic. You need to find a pathogenic mutation. And so the methylation will let you know whether or not this is a pathogenic mutation or benign. So David Putty got an result of FSHD2. It is not a genetic result. It's a research test epigenetic result. It means that both chromosome 4s are epigenetically dysregulated. They're both unmethylated, as indicated by the yellow. Okay? And at least one of them, not both, but at least one of them is a 4QA or 4QAL. All right? It's an epigenetic diagnosis, epigenetic research test. It's not a clinical diagnosis. It's an epigenetic result, a research test result. Okay, now you can have a result that says FSHD2 when you're not FSHD2. These are some of those in the statistics I showed at the beginning where we get a little bit wrong. You could have two short contracted chromosome 4s, and this would look like FSHD, FSHD2, right? You have this short one is contracted, hypomethylated, but so is this one contracted and hypomethylated. Technically, this is actually FSHD1. Compound FSHD1. It's not FSHD2 unless you have an SMCHD1 mutation. So sometimes we get it wrong, but in the end, it's FSHD. It's only going to affect inheritance. Right? Still FSHD. So what does a result of FSHD1 mosaic mean? Now it's turning out this is coming up more and more common, and the stats are starting to show that potentially up to 30 to 50% of the first person that's FSHD in a family is actually mosaic. And they're often unaffected or so mild they don't even notice it. And you pick it up when more than one child is affected. More than one child has FSHD. One of the parents is FSHD. And if they are perfectly unaffected and clinically non-FSHD, chances are they're mosaic. So what are we talking about here? Because our testing picks it up. Okay, mosaic, remember you have two chromosome 4s, one from mom and one from dad. But one of the chromosome 4s could exist in two genetic variants. If the mutation happens, after you get the chromosome, right after the sperm or the egg is generated, then you could actually have one fraction of the chromosomes are non-contracted, the other are contracted. Okay, epigenetically, what does it look like? Because again, we're not doing genetics, we're doing epigenetics. It means that, well, your methylated non-contracted one, that's 50% of what we're assaying. The other one should be 50%, but because it exists in two forms, for example, it could be 12.5 to 25%, depending on where the mutation happened, 2-cell or the 4-cell stage, or even the 8-cell stage. And so you get this skewing where you have more than 50% are heavily methylated and less than 50% are unmethylated. This is epigenetic mosaicism. Okay, so Sally Weaver came back with a result of FSHD1 mosaic. It means epigenetic mosaicism, unmethylated at like 12.5%, methylated, here we are all the way up at 87.5. Means this was actually a mosaicism that happened probably at the 4-cell stage. And so this is what we're doing. It's an epigenetic result. So how does this happen? Here's the picture of kind of what you should envision. Now remember, one chromosome 4 came from mom, one came from dad, one came from the sperm, one came from the egg. Okay, so you get one chromosome and you become a 1-cell, okay, little 1-cell little dude or little dudette, and then suddenly it divides and you are 2 cells old. Now if at this 2-cell stage the mutation for FSHD happens, well, one of your cells does not have the mutation, the other cell does. And so you have one FSHD cell and one non-FSHD cell. Now these divide, you have 2 and 2. So now you basically start going through development where you are 50% genetically FSHD and 50% genetically not FSHD. Okay? And so this also, now it's just a matter, not even just a matter of, a rare amount of cells being on. You are actually half of you is genetically not FSHD. And so this is often very mild, and this can only happen with the first person in the family, because if this individual passes on to the next generation, either all the sperm will be, be FSHD or not, and you can't pass on mosaicism. All right, if you pass on FSHD, your child is 100% FSHD. If you pass on the healthy one, they're 100% healthy. That's just how, in respect to FSHD. All right, so that's what we're talking about. Now, it does not matter if you're FSHD1, 2, or mosaic. In the epigenetic research test result, you will get the following statement. Having the epigenetic characteristics for FSHD, and thus likely the genetic characteristics for FSHD, does not mean that you have clinical FSHD or that you will develop clinical FSHD. There's great variability in the age of onset, clinical presentation, and progression of FSHD symptoms among individuals, within families, and among certain racial populations. If you do not currently have muscle weakness, you may or may not develop symptoms of FSHD in the future. Many individuals that meet the epigenetic and genetic criteria for FSHD are asymptomatic or only mildly affected for much of their lives. If you start experiencing muscle weakness, pain, or related issues, this could be due to emerging FSHD presentation. This should be considered in your management studies. All right, so before you go and have a surgery done, if you knew that you were positive on the research test, you might want to get checked out by a CLIA approved test, a proper, a clinically relevant test before, and make sure that what you are experiencing in whatever's wrong with you, your pain or your muscle weakness, make sure that is that the FSHD or not, because an unnecessary surgery could just make things worse. Okay, so what does a result of not FSHD mean? Okay, well, again, traditional FSHD1 testing measures the size of the array. So genetic testing would mean that you have greater than 10 repeat units on both chromosome 4s, or at least all the permissive chromosome 4s have greater than 10 repeat units. You could also just be BB, in which case this is also genetically not FSHD, and it doesn't matter the size of your repeat units. But this is genetic result that you get. Epigenetic just means that all of your permissive D4Z4 arrays are epigenetically off. They are silenced and they are heavily methylated. Okay? That's epigenetic. That's the result that we give you, not FSHD. So Jackie Childs, Jackie's fortunate, has an epigenetic result of not FSHD. Okay? Now, it does not matter what the distal units are, permissive or non-permissive. The key is anyone that is permissive is epigenetically off. All right? How about Susan Ross? Well, Susan actually is not FSHD, but it's a little bit different kind of not FSHD because, she is not FSHD because she's BB. And it doesn't even matter that one of them is epigenetically on, because the key is you can't have FSHD if you're BB, so we know that she's not FSHD. So this is more of a genetic, we do this little bit of genetics, still a research test, but this is a genetic not FSHD that we do actually be able to put out on a research basis. Okay? BB, never FSHD. All right, so I get this question all the time. Well, why doesn't it say healthy? You said I'm not FSHD, but why not healthy? I got no idea if you're healthy. All we are checking is if your sample in respect to FSHD epigenetics. We do not know if you are healthy or not. All right, so Babu Bhat here got the result of not FSHD, but if Babu is clinically diagnosed with FSHD or a neuromuscular disease, then what this basically says, I don't want to say that, gee, you're healthy, because actually what it really means is you have a different neuromuscular disease, just not FSHD, because your result is not consistent with FSHD. You might still want to go get this checked with a clinically relevant FSHD test, but chances are you want to go on and do another type of analysis. All right, so what might you want to do? Well, first off, get together with a genetic counselor and your neuromuscular disease specialist and to discuss your options. Okay, you can't stress enough how important genetic counselors are in all of this whole process. Unfortunately, I can't pay them. They deserve to be paid for their service, and do what you can. Contact them, and hopefully your insurance will cover them. But a genetic counselor, if you can talk to them, is fantastic, even about the FSHD testing and everything, but definitely about if you're clinically affected and negative for FSHD, you may want to do a neuromuscular disease gene panel and or exome sequencing, but talk with them about what you got. There are other specific like myotonic dystrophy tests, SMA tests, number of things to do. Talk to your genetic counselor. All right, so what about carriers? I often get this question: can you be a carrier of FSHD? So FSHD1 genetically, there are no real carriers. I mean, you can think you're asymptomatic or mildly asymptomatic, but FSHD2 there is a real carrier situation, right? FSHD2 is a combination of a permissive D4Z4, a certain size array, and a mutation in the SMCHD1 gene, most often. All right. So if you're BB, you cannot have FSHD 2, right? This cannot be FSHD. Can't be 1 or 2. But if you're dysregulated on both arrays, that indicates that you do have an SMCHD1 mutation that's likely pathogenic. In which case, you can pass that mutation onto your kids. So they can be an epigenetic healthy carrier of FSHD2, and our testing does determine that on a research basis. Okay? It can be passed, so that's why it's important to know. Right? If FSHD2 is in your family and you're feeling fine, everything's good, you might still want to get checked out to see if you're a carrier, just to see the risk of passing it on to the next generation. All right, regardless of the results, you can always contact me with questions. I am the person you deal with, Peter Jones. Okay, contact me. Occasionally I pick up my phone. Oftentimes I'm very busy. Give me an email, peterjones@med.unr.edu, and I will set up a time to have a Zoom call or a phone call. Prefer Zoom calls, and we will go over it as soon as I can. Any questions you have. All right. So then, section 1, just going to go right through what are the sections. What does it look like? Overall narrative. This report represents the results of FSHD research test analysis for you. Indicates you are epigenetically FSHD type 1. Okay. There is a very strong correlation between epigenetic FSHD and genetic. But these are two important things. It's a research test, and it's an epigenetic test and not a genetic test, okay? These are very important, right? And this will be in everybody's report, regardless of your result, kind of tailored to your result. If FSHD1, then we put in the currently accepted definition of FSHD1, okay? The genetic definition. If you are epigenetically FSHD2, we tell you the current definition of FSHD2. If you're not FSHD, then the currently accepted data, just know it's very clear. These are the official definitions of these conditions, and how this relates to the epigenetics. Okay, I point out that we have diagrams at the end. I tell you again to give me a call. I point you to the MyFSHD website, and then I tell you it's a pretty darn accurate test. But you know what? It does not currently constitute a medical diagnosis or official genetic diagnosis of FSHD. It is still recommended that all participants undergo clinically approved genetic testing for confirmation of this result. Cannot stress that enough. All right. So if you're FSHD1, you might have this additional information. If multiple members of your family, might have, in addition, there's FSHD1 in your family, and genetic analysis confirms that you did inherit the familial FSHD pathogenic chromosome. How do we do that? Well, I told you about some haplotyping in the previous section, but basically there are these little variable regions of the genome. It's kind of like crime scene tracking. We can actually track chromosomes through a family tree. And if we know that the proband here came back epigenetically FSHD, as they did, and we know the DNA markers for this chromosome, we can confirm results, right? So if you're looking at a pedigree, so dad here reports FSHD-like symptoms, but has never been genetically tested. That's what this half and half means. Well, the proband, who was properly genetically tested, also participated. Actually, you can see they inherited the same chromosome. They share the same chromosome 4 with dad. This unaffected sister came back positive as well. Now we can confirm our epigenetic result by saying, isn't it interesting? She also inherited the same chromosome from dad as the affected and confirmed sister. This sister came back negative, BB, and we can confirm that she did not inherit the pathogenic chromosome from dad and does not share the pathogenic chromosome with the two sisters. And then the brother here came back positive, epigenetically positive, and we can again show that they also possess the same chromosome and inherited the same. So we're able to back up the epigenetic results by tracking the chromosome. And then, of course, this individual here, without even knowing that she was going to be positive, did have the children tested and knows right away that both children inherited the non-FSHD4B from mom, and so it didn't get passed on anyway. So this is a pedigree and the kind of thing that we can provide if everybody is willing to participate and share their results with the family. And it's an extra level of confirmation of the epigenetic results because you should have the same chromosome. Now there's some recombinations, some funky things that happen, but we go over that all with you. All right, section 2 is haplotyping. This is, 4A161, 4B163. This person has one FSHD permissive chromosome, one non-permissive. Again, permissive, not pathogenic. 75% of people in the world have at least one FSHD permissive chromosome 4. So what if you have chromosome 4AL? Right? I've mentioned this in other sections. All 4AL is a slightly different genetic variation right here. You make a slightly different DUX4 mRNA transcript, although it's the same protein, and it has a slightly different sequence that is being analyzed by our epigenetic test. So we have to do a separate 4AL epigenetic test. It is still permissive with the polyadenylation signal for DUX4. Okay, so 4A and 4AL, really there's no real difference, just different metrics for studying it. We have learned that 4AL have very high methylation compared to 4A in the FSHD state. So we have to use different metrics for the determination of FSHD versus not FSHD. Okay? There's also something called 4A166. Now I listed here as not associated with FSHD. I really should say not typically associated with FSHD. It is technically permissive. So it does have the PAS. Being permissive or not permissive just means you have the DUX4 polyadenylation signal in the A subtelomere. But 4A166 has several other changes in the sequence, and I'm not entirely sure where they all are, but it has been published by the Silver van der Merel lab and Richard Lemmers in a Human Molecular Genetics paper that contracted A166 does not track with disease. Minimally, it's less penetrant. If it does cause disease, it's going to be a much milder form. And so we just have to be very careful when we look at A166 and we analyze it separately. All right. So 4B, you can't be FSHD. Your haplotypes are either 163 or 168 or a combination of the two. Non-permissive, you don't have FSHD, genetic FSHD, and you can't pass it on to your kids. Again, research test result, have it confirmed, but this is actually a genetic result right here, not epigenetic. And again, that is because you don't have the polyadenylation signal for DUX4. FSHD is caused by the pathogenic expression of the DUX4 gene in skeletal muscle. In order for this gene to be transcribed into a stable mRNA that will be made into protein, you need to have this sequence here called a polyadenylation signal in exon 3 on the 4A sequence that leads to the string of A's being put on. This is required for this message to be stabilized and translated into protein. You need this for FSHD 1 and 2. If you're 4B, there is no signal here, there is no polyadenylation, this RNA is degraded, no DUX4 protein, you don't get FSHD. So this is what we mean by permissive and non-permissive. Okay, so section 3 will be the first time we're going to look at, are you FSHD or not FSHD? Remember epigenetics, we're really looking at off, on, on. Okay, we're only going to look at permissive chromosomes, so we don't care about B, we only look at the permissive ones. So we're only going to look at these. All right, and we're just going to see, is there any population that is low levels of methylation? And the answer is yes. Then we'd say you're epigenetically FSHD. And if not, the answer is no. We'd say that you are epigenetically not FSHD. Okay, if you're 4A, we're looking at both chromosomes. Okay, we're going to look at Q1, first quartile, 16.7. 16.7 is your relevant quartile. It changes whether you're AA or AB or AAL. Okay, and that is FSHD. If you're AB, okay, quartile 2 is your relevant quartile. That's right here, and that is going to be FSHD as well because it's, on less than 26%. All right, if you're 4A, 4AL, here's an example of the different metrics. This 4AL, that's right, so 4A, this is a healthy 4A, so it says not FSHD. Now I get confused and say, but wait, it says I'm not FSHD. No, it says your 4A chromosome is not FSHD, but you also have a 4AL chromosome. And the first quartile is 33.3%, which you say, wow, that's higher than, FSHD levels. Not for chromosome 4AL. That's right in the wheelhouse of 4AL. We're then going to look at the 5th percentile and the 1st percentile. This is a very standard FSHD signature for 4AL. The numbers are just higher for reasons we don't understand in the FSHD situation. All right. So what about mosaic? We mentioned some people are mosaic. And this is actually becoming more and more common, and this is going to get missed by other testing. I think that BioNano is probably pretty good at testing it, but other epigenetic analyses that people are proposing are going to miss this, and Southern blotting often misses this. And so anyway, it's very important to know for family planning. And also some of these people are affected, mildly affected, right? So we're going to look at this distal most repeat, right? And instead of a 50, if you're AA, you should be 50% methylated, 50% unmethylated, but the percentages are going to be skewed because one chromosome 4 exists in two populations, right? So you get a result like this. Here's a 4AB result. So initially we would just say, okay, your AB is second quartile, not FSHD. But then I look at it, and again, I am the one looking at it, and I'd say, whoa, wait a minute. 5th percentile is 0. We never see that in someone that's not FSHD. 10th percentile, 4.2. No way, this is definitely FSHD. You only ever see these in FSHD cases. And so, but it's skewed. So this suggests mosaicism. Okay? FSHD. All right. What does it look like when we see the heat map? Now remember, blue means unmethylated and red means methylated. They're AB. So everything here is, this should be one genetic population. And we understand there's epigenetic variability, but this is a very striking epigenetic variability right here. What we see is 25% is FSHD. So likely there was a mosaic mutation at the 4-cell stage, okay, and led to this person being 25% FSHD, 75% not FSHD. That's a real mosaic, and that's going to get missed by these PCR assays that just look at overall methylation states. And that's really important because I got to tell you, this person likely is affected. In fact, I actually know this person is affected, because look how I mean, they're like a one repeat in the background of, you know, a much larger array. But they're not infantile. Normally that'd be infantile, but they're older and they're affected because, you know, so it's important to get this right. And of course they're going to pass this on to their kids, and their kids, they could have multiple very affected kids in this case. All right. So section 3 is determination of FSHD. What if it's not FSHD? Well, that just means, you know, you're very heavily methylated. Clearly, all right. So if you're 4A, again, the first quartile, 4A is always going to be the first quartile. Clearly, you can see the metrics are right here. Greater than 35%, not FSHD. AB, it's going to be the second quartile, relevant quartile, greater than 35%, not FSHD. AAL, both of them are going to say not FSHD. Now you can see the very different, these are very high numbers for the L, not FSHD. And first percentile is always less than 5% in FSHD. Fifth percentile is always less than 12.5% actually. So there you go. So if you're BB, it'll say not applicable. Okay, not FSHD. All right, this assay only works on A and AL. We still run the assays to make sure you're not a rare haplotype. Make sure it's not something's recombination event. Make sure there are no mixing up of samples. Honestly, we just ran this on everybody. Okay, not applicable. So then section 4 is going to be, is it FSHD2 or are you a carrier of FSHD2, right? We're going to look at all of these D4s, Z4s, all of these, all of these, all of these, and right, and do you have more yellow than black? That's really the question. Unmethylated versus methylated. Okay, and then are you a carrier? Well, here's someone that's positive for FSHD2, okay, and they have a permissive allele. They would be FSHD2. They're going to make DUX4. But this individual similarly is positive for FSHD2, but they're BB, so they're going to be DUX4 negative and no FSHD, but they would be a carrier of FSHD2 and could pass FSHD2 on. Okay? So, is it FSHD2 or are you a carrier? Basically, if you were positive for FSHD in section 3 and not FSHD in this section, you most likely are FSHD1. Okay? There we go. If you were positive for FSHD in section 3 and FSHD2 in section 4, then you are most likely FSHD2. Okay. If you are BB in section 3 and FSHD2 in section 4, then you are most likely a healthy carrier of FSHD2. Okay? And if you are not FSHD in section 3, and it's not because you're BB, you're going to be not FSHD here, but we check you anyway. Everyone gets a result here. Lets us know that your DNA is fine, the bisulfite conversion worked fine. It's just another check and balance of the whole system. Okay, section 5 then will be this long report where I write everything. The key is going to be paragraph 2. Paragraph 2 is where I go through the numbers. I would fill in your numbers here where I have X's, and I will tell you how you met the criteria that we came to our conclusion. All right? So I also put in further interpretation. This is where I explain the idea that we get DNA from thousands of cells from your cheek. All right? They all have the same genetics, but they have different epigenetics. And I just kind of go through the complexity of FSHD. I do point out that, we're doing our best, and our understanding does change. Actually, if you took our testing 4 years ago, we might have slightly different results now because we have a better understanding of the cutoffs and the mosaicism. And I'm reaching out and contacting a few people to let them know that actually they weren't negative, they were mosaic, back in the early days. Not FSHD, the same sort of thing. It doesn't matter whether you're FSHD or not. It's paragraph 2 is the key paragraph here. We do make sure that everyone knows this is a research test result, and we really stress again to undergo clinically approved testing and consultation with a neurologist or genetic counselor to confirm these results. Okay, afterwards there's a disclaimer to cover our ass, but it just tells you, again, it's a research test, and we're doing our best, and this is based on our best understanding. We are a research lab. We're not a CLIA-certified lab. We're not pretending to be a CLIA-certified lab, and, mistakes happen. Very rarely, but it's possible, okay? Here's the interpretation. You can actually go through and read about the BSSA assay, the L assay 166, BSSX, and kind of write it out how we do things. I do point out that molecular genetic testing is truly the way to go. You want to get a CLIA certified diagnostic test. Where can you get them? I actually highly recommend the University of Iowa Diagnostic Laboratories. I think they're the best in the U.S., but there's other ones you can go to. And then genetic counseling. Genetic counselors are fantastic. We work with a number of them. Really valuable expertise. If you have an opportunity, definitely discuss everything with a genetic counselor. All right. Now, I will point out that you can get a conflicting result, right? Sometimes, again, people get a result from us that says epigenetic FSHD. They go and they get their clinically approved test, and it says not genetic FSHD1, not FSHD2. And then they get all like, Jones, you told me I was FSHD. I said, oh, it was a research test. You are epigenetically FSHD. I'm not telling you anything, really. Just your results are consistent with epigenetic FSHD. But what's going on? Well, oftentimes we will find that either they have a contracted 10, which is could be, see the first section on the genetics if you didn't see it. You could have a contracted 10 that is recombined with the 4, and genetic testing gets that wrong. But also sometimes I said, can you send me your results? Hopefully they'll tell me the size of your arrays. And it's not that uncommon to find someone's got an 11 or 12 repeat units, and that's not considered FSHD. So you could be epigenetically FSHD, clinically FSHD, but if you have 11 repeat units, you don't fit the criteria for FSHD. And then they tell me my testing's wrong. No, my testing's right. Our testing is right. Our research test is accurate. It's the definition that needs to be changed. Anyway, I'm happy to talk to you about it. It still should be considered FSHD. You know what? Some medical doctors, quite honestly, they want what's best for their patients. They know they're clinically FSHD. They know they're epigenetically FSHD, and they know the definition's wrong. I think some of them are doing a solid to their patients and telling them, you know what, you're FSHD. Now, at the very end, I'm going to have a series of diagrams that cover some just genetics, basically chromosomes, go through the two types of FSHD, FSHD1, where the signal to be turned off is broken, FSHD2, where the switch to turn off is broken. I then explained the testing that I'm just talking about here. These are all in your reports. Go to the back end of your reports. That's why your report is so long. It tells you this is the first test we do, then the second test is right here. We do this test, epigenetic assay 2. And then also I explain how you get the different quartiles and how the quartiles are set up, whether you're AB or AA, FSHD1 or not FSHD. Check it out. Good information. Hopefully it's helpful. You know I talk too much, but you know what? I want it there for you. I'd rather you have too much information than not enough information.